Okay. Hi, Alan. Thank you very much for agreeing to give a Build a Cell seminar, and you can get started.
Wonderful. Well, thank you so much, Kate, for the invitation to present here. This is my second time presenting for the Build a Cell summer series, and really excited to share with you some of the recent work that we've done, and try to organize it in a way that hopefully tells the story on our effort towards building a force-responsive secretory synthetic cells. So like many people in the field, we're interested in building synthetic cells as I see cells as a basic computational unit. And the idea is that we can build a synthetic system that emulate this input and output relationship that cells are able to exhibit. So if you look at a natural system, cells are able to sense a variety of inputs such as forces, molecules, or light. And then through receptor signaling and other mechanisms, they're able to produce forces and allow them to change shapes and allow them to have a variety of enzymatic activity or biochemical activities. And some cells are able to secrete. So our synthetic cell models are pretty straightforward, and which is a lipid bilayer vesicle that encapsulates biomolecules, and we also try to endow membrane properties to them by expressing receptors or membrane channels. So in my lab, we use synthetic cells for two primary purposes. One is we use them as a model system for cell mechanics, and the second is we use synthetic cells as a tool for understanding cell-cell communications and be able to use them as a conduit for communicating between a synthetic cell system to natural cells. So the motivations that at least I have from going to this field more than a dozen years ago is motivated by a perspective article from Petra Schwele, who described the use of molecules as a building block for synthesizing or creating a synthetic cell system. And here, illustrated here is a variety of molecules that people have studied for many years now. And the idea is that can we take these components and build a cell, right? So this is the area of cell reconstitution, where we can assemble biological components in a meaningful way to build a synthetic cell that will mimic some aspect of a cell. So this area of research has grown over the past decade or so. And we not only can use it for understanding biology, but we can also use it for practical applications in say, biomedical fields or for in the environmental field. So I want to motivate my interests of building in synthetic cells, which essentially my lab is broadly interested in how cells can sense, respond, and generate forces. And here's a video capture in the 1950s by David Rogers that described a neutrophil that chased after a bacterium. And you can hopefully appreciate the fact that this neutrophil has to be able to sense the presence of bacterium and chase after it by generating forces, protruding a leading edge, and consistently go after this bacterium and eventually ingest it. So this highlights the ability for cells to have this autonomous movement. And the second video highlights the ability for cells to communicate at membrane-membrane interface. And this is a video showing a killer T cell that when in contact with the ovarian cancer cells, secretes some kind of toxin component and basically kills these cancer cells. So this is a hallmark of biological system that cells are able to communicate when they're in close contact. So with that, I wanted to sort of discuss our lab's recent research in the context of our long-term goal, which is a ongoing endeavor that we want to be able to engineer robust and autonomous migrating synthetic cells. They're able to communicate with living cells via physical and biochemical interactions. So my talk will be broken down into three vignettes. First on cell skeleton reconstitution, second on membrane interface engineering, and I'll finish off with synthetic cell secretions and then try to give you a sense of how these are connected to achieve this long-term goal. So I'll start with cell skeleton reconstitution. Okay, so my lab has a long-standing interest in the actin cell skeleton, and these are the molecules that, when polymerized, are able to produce the forces that drives the cell to migrate. So as you saw in the video earlier, the cells are able to migrate because of these monomer to filament transitions, and then be able to protrude against the membrane. And this is also the mechanism by which the cells acquire its mechanical rigidity. Importantly, these actin filaments, when polymerized, can interact with a range of accessory proteins that essentially provide different functionality. The one we're going to focus on include cross-linking proteins. So they allow these filaments to be cross-linked. Bundling proteins that allow these filaments to be bundled into a into something more rigid. And then my lab has also worked with motor proteins, which I actually won't talk about today. So they allow the filament to slide against one another. For the purpose of this talk, all you need to know is two type of actin architecture, right? So at the leading edge of a cell, there are branch network that essentially give rise to this lemmapodia. And these are the dendritic actin network that forms a broad protrusion front for a cell. And there's also the bundle actin network that forms these more rigid protrusions that are like finger-like protrusions that are called filopodia. So the actin filaments can be modified depending on the binding proteins to take on these different architecture. So the two specific actin binding proteins that I'm going to first describe to you are FASN, which is a short cross-linker, and then alpha-actin, which is a long cross-linker. It's been shown previously that when you take actin filaments together and reconstitute with two different actin binding proteins, you will create a domain that has short cross-linker and a domain with long cross-linker. And the reason that's pretty simple is there's entropic penalty for having these filaments, so I guess the filaments like to be clustered together so that you don't have these wavy bending portions of the filament. So the long cross-linker would tend to be formed in one domain, and the short cross-linker would be the other domain. So how do we build these synthetic cells? And probably many of you have heard of this technique. We use a variety of techniques in the lab, and for this particular studies, we've been using CDICE, which stands for continuous droplet interface cross-encapsulation. So it started with we emulsify the proteins that we want to encapsulate, which includes actin and actin binding proteins. By taking these emulsion that are in the presence of a lipid, we can then send it through this rotating chamber. These emulsion droplets will acquire a second monolayer and forming a lipid bilayer vesicle. And this is an image of actin that's crossing by probably perhaps alpha-actinin, and you can see these label actin, that are encapsulated within a spherical vesicle. So this strategy gives us very high encapsulation efficiency, but we do have a variety of sizes. It's very easy to do, and then if we load these vesicle in a 96-well plate, we can image them pretty readily after it's formed. So the first experiment I'm going to show you is simply encapsulating actin with this short cross-linker FASN I talk about. So these FASN will basically create a bundle actin that are very rigid, and these are found in the filopodia that protrudes at the leading edge. So if you encapsulate this, you can see that in the case here that these bundle actin protrude outside of the giant vesicle, and essentially looks very much like a cell. Now, we can use light to essentially, with very strong exposure light, we can actually depolymerize the actin. And you can see that as the actin is depolymerized, the vesicle becomes spherical. And this suggests that the protruding actin is what, the actin bundle is what is deforming the giant vesicles. So what happens if you include another cross-linker alpha-actin? So the cell skeleton can self-assemble on its own with a variety of actin cross-linker. And when we incorporate, so if this is alpha-actin alone without FASN, right? So the alpha-actin is a longer cross-linker and creates these rings when the vesicles are small, and we can tune the ratios of alpha-actin to actin. As we increase, look at the larger size of the vesicle, you start to see sort of more random network that form in these vesicles. Now, if we include alpha-actin and FASN together, so having both a cross-linker in the same system, we begin to see something interesting. We begin to observe these astro-like structures that are highlighted in these arrow here. And if you look at one example of this, you see a domain that is fairly compact, and you have these bundle that radiate outward. So this is a complete due to self-organization. If you take the same proteins and mix them together in a bulk system, you do not observe these astro-like structures. So it suggests that the boundary, the confinement actually can provide some type of organization for this two cross-linker system. We perform fluorescence microscopy of label alpha-actinin, label FASN, and also along with label actin. And hopefully you can appreciate this is the alpha-actinin. You have a very, very dense alpha-actinin cluster at the middle. And then if you look at the FASN channels, you see the FASNs are labeled along these radiating bundles, and this is the actin channel. If you do a line scan across the sort of circular regions, you will see that the alpha-actinin will have these spikes. and the alpha actin, sorry, so the FASN has these spikes. Sorry for that. The alpha actin is more or less flat. If you look at the line scan across, and you see the alpha actin has a very pronounced signal in the middle compared to FASN. So what that's telling us is that these proteins will segregate on their own in very similar to the two filament system I showed you earlier. Is that they're trying to minimize the bending energy, and here they can also do the same in the confined system. In collaboration with Aaron Dinner's lab at University of Chicago, we performed some cross-screen modeling of the system. And if you look at the alpha actinin simulation and the FASN simulation, when you only contain a single component, you don't see much of a feature for the two. Now, if you combine these two cross-linker in the same simulation, and you'll start to see that alpha actinin is enriched in the middle. And those signals here is really poor, but FASN will sort of more be more devoided in the middle and occupy sort of the external space. So this sort of simulation also confirmed what we observed experimentally, which is that the alpha actinin cross-link portions are in the center, and then you have the FASN bundle stiff film bundles that are radiating from this alpha actin rich domain. So what the study show you is that the cell skeleton reconstitution can provide a platform for looking at multiple cross-linker competitions, and we've seen this in other scenario as well, with different type of actin being utilized by the same pool actin in converting and essentially competing against one another, in organizing a structure within the cell. So I mentioned earlier that actin is used for force generation, but it's also very important for stabilizing, or I guess providing the mechanical rigidity of a cell. So in this context, we encapsulate actin the same way that we did before, and my student Dab, develop a electrodeformation assay to look at the deformability of a giant vesicle encapsulating actin. So idea is that we can pass a alternating electric field and look at the long axis to the short axis ratio as a measure of deformability. And just to give you a glimpse of what this looked like, if you encapsulate F buffer and you will have a rather large deformations with this A to B ratios of 1.45. If you encapsulate F actin, you can see that the deformation is slightly dampened, but you can still see, see the deformation. Now, however, if you form a cortex, and this is mediated by formations of a cross-link, or I guess a branched actin network that form at the membrane. Here we use 5 micromolar actin. You can form a very nice thin cortex at the membrane surface, or I guess in the inner surface, and you can see that the vesicle barely form under electric field. Now, if you cross-link the actin using alpha actinin that I introduced earlier, again at 5 micromolar actin, and this you can see that the vesicle can be deformed by electrodeformation. So summarizing over here is that if you form a cortex, then this deformation is significantly dampened compared to a cross-link case. So I think here it highlights a very important message that depending on the organizations of actin, the cells will can regulate its mechanical behavior, and this is can be illustrated by this sort of idea that here you have toothpicks that are arranged in a structure that can actually support a great amount of load with certain block. This truss structure is known to be very stable against compressive stress. Now, if you build a structure in a different way, then it may or may not be able to support this weight. So the cells is able to rapidly alter its mechanical behavior by rearranging the actin filaments in a structure that either support force generations or maintain its rigidity, so it's able to move in a very malleable fashion. So the last little sort of want to share here with the cell skeleton reconstitution part is how does the cell skeleton or actin cell skeleton respond in response to forces. So here my student Najab did a pipette aspiration assay where he encapsulated either actin bundle or actin bundle that are branched using R33 complex, the same branching protein found in Lematodia. And then he looked at how they're able to respond to micropipette aspiration, so the force they're exerted by suction. So here What he found is that if you included just bundle actin inside the vesicle, as you aspirate on the vesicle, hopefully you can appreciate see that these bundles begin to align, right? They begin to reorient themselves in the directions of the aspirations, and then you can actually suck in a rather large bundle into the pipette. And this is also, useful in the context of how cells are able to remodel their cell skeleton in order to generate forces towards a certain directions or be able to, say, be softer because they're not cross-linked and be able to flow into a small space. Now, in contrast, when these bundles are cross-linked, they're actually a lot more rigid. So as you aspirate on the vesicle here, these cross-linked or these branch bundles barely deform and basically resist the suction to a greater extent. So that sort of completes the first. In the end, is that we have been able to build these actin cell skeleton vesicles as a model for cell mechanics study, and that is important for to be able to eventually create this autonomous migrating cell. This is a long-term goal of ours. The second VM I'm going to talk about is membrane interface engineering, and this has to do with the motivation I showed you earlier that cells are able to communicate through a membrane interface, and this is work done by Hossein. So several years ago, we came up with a system to modify membranes, two different membranes, and have them reconstitute a enzyme or a protein in between the membrane interface. And we call this inner spy. And what this is a transmembrane helix here from the transferrin receptor. We link into a GAP. And then we utilize spike catcher and spike tag interactions to create a covalent bond. So spike catcher and spike tag come from this fibronectin binding domain that is modified so that it can form a covalent bond. On one hand, we have SF cherry 1 to 10. So this is a incomplete SF cherry, and then on the other protein fragment, we have the SF cherry 11. So when the two come together in close proximity, it can reconstitute fluorescent SF cherry. So this other component has a PDGFR beta transmembrane helix with BAP for marking it, and then the spike tag to interact with the spike catcher. So when we reconstitute this, the expectations that you will see the SF cherry in this complemented protein. So this this experiment is rather simple. So we made a super template. So these are small univesical. Super templates are supported by layer with excess membrane reservoir, and then we deposited on them SUVs that contain one of the partners. So here is an illustration that if the SUVs are able to bind to the other partners on the super template, then we'll be able to see SF cherry. So we use for this experiment, we use purified protein that can bind to the SUV, and then in the super template, we use mammalian self-expression to incorporate the other component. So hopefully you can see that the super template is on essentially a glass bead, and you can see that the BAP is there, the GAP is there, and then you also very nicely see the SF cherry, suggesting that the two complements of the inner spy are there in order to reconstitute SF cherry. We can reverse the protein partners on the two membrane, and we again see the formations of SF cherry, right? So BAP on the membrane, GAP on the so the BAP here is now on the SUVs, the GAP is on the super template, and then we also see SF cherry here. I should just point out that if you utilize the component that does not have a spike tag, then you will not get any SF cherry reconstitution. This is highly dependent on the complementations of spike tag, spike catcher in order to reconstitute SF cherry. So this is great. We're able to reconstitute a split fluorescent protein at the membrane interface. The question is, how can we use it to entail a function between synthetic cells and be able to allow synthetic cells to communicate with one another. For this, we decided to use light as a primary signal for synthetic cell communication. And part of the reason for using light is it doesn't exist in natural systems. So this is almost like bringing a new functionality that the cell don't use. But as synthetic biologists, we can essentially create any type of signal that we want as long as we can form a way for this communication to pass down the information. So it has two parts. One part is signal generation. So light can be produced by luciferase, and here we essentially tag spike tag with SM bit and spike catcher with LG bit, and LG bit and SM bit will complement and form nanobit, and so this is a way to generate blue light with the single generation. So when the spike tag spike catcher come together, we can generate light. The receiver side, we propose to use eyelid and SSPB that essentially can bind to one another under blue light stimulation. So, eyelid has this J alpha helix that basically gets exposed to under blue light, and then it will bind very tightly to SSPB. So the experiment is rather simple. We have a population of center cells. We have a population of receiver cells. The center cells will have the spike catcher. The receiver cells will have the spike tag. When they form a membrane interface, we will complement, the small bit and LG bit in forming a nanobit. And the generated light will then drive an optogenetic reaction to bring the luminal SSPB and cherry to the membrane that has the eyelid. Okay. And the study utilizes all purified proteins and no self-re-expressed protein here. So we essentially just had to replace our M cherry that I talked about earlier with LG bit and small bit. And the schematic looks something like this. And we first wanted to test that this can indeed generate light. So we ran a luciferase assay, some nanoglow assay, and you can see only in the conditions where we have the spike tag with SM bit and then the small catcher with LG bit do we get a strong bioluminescent signal. All the other conditions are completely dark. And we also can see that there's complementations with the forming the complex when we have spike tag, and you see this band right here. And then if you without spike tag and you do not form a complex here. Okay, so this is the spike tag SM bit, this is the spike catcher LG bit, and then this is adding the two together. So now that we know that these two things can come together and form a complex and it can generate light, our next task is to show that the eyelid and SSPP work, and this has been very, very well characterized, and you can certainly test this by just illuminating blue light. So for this experiment, we encapsulated M-cherry SSPB inside, and then actually didn't draw here, but there is eyelid that's his tag that binds to the inner membrane. Okay, so if you have SSPB and this is just what SSPB look like. If you have SSPB plus eyelid in the dark, and you just see that the fluorescence is still luminal. If you expose this with blue light, you'll see the translocations of SSPB to the membrane. And this can be quantified over here, and then you can see a very strong membrane to lumen fluorescence signal when you have light, when you have blue light excited. So the next question is, this is obviously expected, illuminating blue light can drive eyelid SSPB interactions. The question is, can nanoluc drive this interactions on the membrane? For this, basically, is the experiment that I introduced earlier is the center cell receiver cell system with each of the binding partners and then generating the light in between and then allowing this to translocate. Now, during developing the system, we realized that we needed to osmotically deflate the cells in order to generate large enough of a membrane contact phase. So you'll see that we would add sodium chloride to create this larger interface. Okay, so the center cells will have spike catcher LG bit. The receiver cell has spike catcher LG bit, and this receiver cell has spike tag SM bit. And then with the eyelid and the lumen, and also SSPB and the lumen. Now this eyelid will bind to the membrane. And then we mix these two, and then we add fluorescein as a substrate to generate light, and then see what happens. So these are the negative controls, right? So in the absence of fluorescein, or if you don't deflate the cells, you see a nice populations of center receiver cells that do not have any feature here. And if you don't have fluorescein and you have sodium chloride, you still don't see a signal. Now, if you have sodium chloride and fluorescein, you can see that there's a signal on the membrane for the SSPB and cherry. And this is quantified over here. Even though the signal is low, there's certainly a observable signal that we can see from the condition of having both sodium chloride and fluorescein. So what's kind of signal, and this is where the sort of work stop is what signaling can contact-dependent light generation drive. And just want to show you an image where, a populations of sender and receiver cells, and you can see that the center cells, in the receiver interface will have some signals of M Cherry, whereas the distant cells do not have that. Okay, so this is sort of mimic that videos I showed you earlier, where the T cell, the killer T cell, interfacing with the cancer cells and are able to kill just the cancer cell next to it, but not the distant cell. So one can imagine as a continuation of this work is to see if we can use this to activate using the blue light generated by this interface to activate a calcium permeable channel rhodopsin. And this is something that has been described in the literature if you can use the blue light to activate a channel rhodopsin, allowing calcium to come in, and maybe that can drive some type of reaction. Another idea is to use the system to drive the translocation of a lipid modifying enzyme, and such as PIP kinase or phosphatases, and that can alter the local membrane composition. And finally, you can also imagine using the system, the optogenetic response to recruit a cell skeleton motor proteins or cross-linker bundle. Okay, so the last portions of the talk want to describe our effort in synthetic cell secretions, and I want to highlight two different systems for secretion. One is to use membrane fusion as idea. So this is two membranes that can fuse together. Second is a relatively new study that we published that basically bridges what we know from before, is encapsulating actin inside a vesicle in a secretion system that respond to a protease signal to release the cargo of interest from within the synthetic cells. And this is work done by Yen-Yu, Samuel, and Mushumi. So membrane fusion is a very well characterized cellular process, and it mostly in cells are driven by proteins known as snares. And you can find them at the membrane for exocytosis, between ER and Golgi transport, and even in endosomal fusions. And it's a very well conserved process both in animal cells and yeast cells. Now, people have done many years of study looking at snare and understanding there's molecular structure determinants that give rise to this bridging of a snare complex, allowing the two membrane to fuse. At the base of it is this idea that you have these helix bundle that can bring the membrane very close together. So people have been able to imitate this by using either peptide or DNA. And so you can use this minimum peptide pair called the K and E peptides, and that can bring these two vesicles together and then allow fusion to occur. Alternatively, you can also use DNA, complementary DNA, to imitate this docking, lipid mixing, and content mixing processes. But what's missing from these previous study is its inability to be, I guess, mediated by calcium. And in fact, the living system actually uses calcium as a way to induce, snare-mediated fusion. So the question we want to ask is, how can we reconstitute this feature in a synthetic exocytosis scheme that can be sort of broadly applied to either DNA or peptide? So we came up with this idea where if we attach a PEG chain onto the membrane, and essentially blocking the two membranes from interacting with one another, and in this example, we're using DNA, but we also have done this with peptide as well, is we can graft complementary peptide on each of the vesicles and allowing the PEG to serve as a shield. Now, we can engineer a cleavage site for this PEG such that right here, and this can be cleaved by calpain-1, which is a calcium-activated protease. Now, if calcium is not absent, then this PEG will stay on the membrane and sort of shielded the vesicle from binding and fusing with one another. If calcium is present, then the protease calpain would be active, liberating the PEG chains and allowing these two membranes to come together and fuse. So first, want to test if this calpain-1 works well with in a calcium-dependent manner. So we generate this calpain FRET reporter. So this is the calpain cleavage sequence, and it's made by a quencher in the 4,4. So in the absence of calcium, there's absolutely no calpain-1 activity. If we have calpain-1 and calcium chloride, you can see a increase in fluorescence, suggesting that the calpain is active. We can also encapsulate this into GVs, and in the GV case, in the absence of calcium, we don't see any fresh signal or I guess fluorescent signal from the EDNs. If we incorporate calcium, we can see very robust fluorescent signal. We can also introduce the calcium externally, and we can do that using ionophores. So in the absence of ionophore, there's no fluorescence. And if you incorporate ionophore in the system, external calcium are able to enter. And this is a case where we already encapsulated calpain-1 and also the calpain Fred reporter, and you can see a pretty robust signal. Next, what we want to do is to test that this is a calcium triggerable membrane fusion. For this, basically, we form our GUVs, and then we add a small, 100 nanometer SUV to it with each have the complementary DNA pieces. And we call this, we have two different sequences in the paper that we have published before, and we just call it A and A prime and B prime. So I'm going to show just the A prime case. If you see the A prime case, so the GV will have one, the SUV will have the other, and you can see the membrane signal pretty well on these GVs. If you take out the A prime sequence on the SUV, then you don't really see much of a signal on the GV membrane. And then we can also show that this interaction is blocked by the additions of the PEG, right? So if we add the PEG, here we're using a 1K PEG in 2K PEG. So you can see that the 1K PEG does not block the interactions with the SUV and the GEV, whereas the 2K PEG effectively block this. So this is sort of a confirmations that these PEG are effective, but you have to have a certain size. And finally, we show that this is calcium triggerable. And if you don't have calcium and you have your GV, the SUV does not interact with the G membrane. And then if you have the calcium chloride, then you can see very strong SUV signal on the GV membrane. Now, this experiment, even though you see a red ring, doesn't necessarily prove that there's mixing of the lipid, right? Because it could be that the SUV are simply just bound to the GV membrane. To test that, there's actually fusion that happened with devices assay where we can introduce nickel NTA lipid from the SUV fusion with the GV membrane, and we encapsulated BAP that has a his tag. So if the nickel NTA lipid gets incorporated, then we will see the binding of BAP shown here. Okay, so in the case of in the absence of calcium chloride, we don't see any SUV bound, and our his-tag BAP remain completely luminal. If we incorporated calcium chloride, we can see the SUVs are now on the GV membrane. But more importantly, is that we do see the translocations of BAP signal to the membrane, suggesting that there's fusion that happened, introducing the nickel NTA lipid to the GV membrane. So I think this is the end of the one mechanism of the secretion. I want to switch gear and talk about an alternative secretion mechanism that brings us back to the actin reconstitution work that I showed you at the beginning. So we came up with this idea of releasing a component by cleaving off actin-bound cargo. So the system essentially incorporates the actin reconstitution I talked about before, and we generated a artificial cross-linker that has a LifeAct domain, which binds the F-actin, a TEF cleavage site, which can be activated by active TEF protease, and a cargo of interest that's linked to a cell-penetrating peptide. We generate we call this TRAP, so it stands for TEF-mediated releasable actin-binding protein. And we generate a delta CPP version, so that version should be retained in the synthetic cells. So if you don't have TEF protease, this is the system, the cargo remains bound to the actin. If you incorporate TEF protease, it will allow the release of the trap cargo because of the CPP. If you remove the CPP from the cargo, then we expect no release from the synthetic cells. So this is a bulk assay where we can in vitro polymerize actin filaments, and just to test that the trap cargo co-localize. So the trap cargo are they're purified, so we can see very nice co-localization between the actin and the trap cargo. And when we add TEF protease, the trap cargo obviously gets released from the actin, and you don't see any fluorescence here. And this is also the case with delta CPP. They bound actin just as well, and they're also able to get release, but there's no membrane in here, so all the cargo will be released into solution. Now, if we the next thing is we want to test if TEP protease can cause this trapped cargo to be released from GEV. So the experiment involved encapsulating TEP protease in the GEV system, and we expect no release if there's no CPP, and release if there's CPP there. And so in the absence of TEP protease, we see colocalizations of our actin in the trapped cargo, just like before, with and without CPP. However, if there is TEP protease included, and you can see the actin is still there, but we see a very, very low signals of the M cherry, suggesting that it gets cleaved off from the actin filaments, is able to leave the synthetic cells here. If you don't have the CPP here, we see more, diffuse M cherry signal as the trapped cargo cannot escape out of the GUVs here. So how do you release TEP protease from a vesicle? So the previous experiment, we just directly add TEP protease as we encapsulate. So here we utilizes light activated TEP release. So we encapsulate TEP protease within SUVs, and these SUVs have a photodegradable lipid, and such that when we illuminate with UV light, we can release the TEP protease from these SUVs, and the TEP protease are able to cleave the trapped cargo and allow the trapped cargo to be released. So this is, using this system. This is in the absence of UV light, and we can see that the trapped M cherry are still very well colocalized with the actin. However, in the presence of UV light, the trapped cargo would, be released from the actin and be able to diffuse out. Okay. So that sort of completes the secretion part, and I kind of want to use the last few minutes to bring things back to talk about. I haven't actually, for the entire talk, I haven't actually talked about activation by force, right? So we've been working on this system for a number of years now, which is a mechanosensitive channel that is found in bacteria. It's called MSCL, so mechanosensitive channel large conductance, and this is a channel that's found in bacteria. It forms a pentamer. It's able to respond to membrane tension, and membrane tension can be produced either by osmotic, hypoosmotic shock, or other ways of deforming a vesicle. And so this is a study that we published a number of years ago now, where we encapsulate a couple gene circuits here, and this circuit basically produces a MREB, which is a homolog of actin. It also produces a MSCL, and this the mechanosensitive channel. So the idea here is that under hypoosmotic conditions, the inducer for the circuit can enter, and thereby producing the MREB that will essentially range on the GEV here. So in the absence of, so here I'm just condensing the key data here. In the hypoosmotic conditions, you can see the GEV label in red here, and then if you don't have osmotic shock, there's no fluorescent MREB produced. If you hypoosmotic shock the GEVs here, IPTG will enter and produce the MREB as you see here. And the MREB self-assemble on the membrane in this case, and you can even see it forms a ring, surrounding the inner surface of the GEV. So this mechanosensitive channel is actually quite versatile, and we have used it in a number of studies previously in cells and in GEVs. And this is how we're thinking of bringing the force sensing things together. But before that, I want to show you some published data that essentially shows that we can use calcium to activate membrane fusion within an MSCL reconstituted GEV. So here we produce, let me just show you this. So we produce EGAP MSCL that's reconstituted in GEVs by using Pure. And we do this from the external side. You can see a very nice ring of EGAP MSCL. We encapsulated SUVs within the GEVs, and this has the full complementations of the DNA fusing system, and we do this with or without calpain. Okay, so without calpain is our control, and then with calpain is our full system. And this is done under isoosmotic conditions. So you don't see any fluorescence on the membrane because the calpain is not active, right? Because there's no calcium inside the GEVs. However, if you subject the GVs in the hypoosmotic conditions. In the absence of calpain, you don't see any fluorescent membrane on the GV. However, if you look at the calpain case, in this case, you see a nice ring for the SUV membrane, which suggests that the calpain is active inside, cleave out the PEG, and allowing the DNA mediated fusion to deliver the SUV membrane to the GV membrane. So this is sort of where we're at with the system. So want to give you a glimpse of what we are thinking of moving towards, and so tie it back to this force sensing synthetic cells that's in my title. So essentially, what we're thinking of is a synthetic cell system where we have SUV that contain some type of fusion machinery. It can be DNA, it can be peptide. We have work on both. But importantly, this is a system that it respond to increase in calcium. Okay, and we'll have MSCL membrane, decorating the synthetic cell membrane. And then in the inside, it's going to be an active calpain because calcium is absent. This will be triggered by a mechanical signal. The trigger is due to increase in membrane tension that activates MSCL. And the activation of MSCL will essentially lead to a calcium influx, which activates calpain. And calpain can then cleave off the PEG that's protecting the SUVs, allowing the SUV then to mediate membrane fusion or interactions with the G membrane. So this is a system like a synthetic exocytosis. Finally, we're actually working very hard on trying to build a system so that these synthetic cells can communicate with living cells, and haven't shown any results there yet, as we're still working on this. Is essentially having these synthetic cells interface with living cells so that we can use the synthetic exocytosis to deliver molecules of interest, right? It can either be molecules that promote growth, or it can be molecules that inhibit growth, in the case of cancer. So it's a versatile system for us to explore. All right, so with that, it's come to the end of my talk. I want to thank my lab and highlighted the folks that actually did the work along the way. I want to mention that Ishar was a past postdoc that did a lot of the actin along with the DAP. Hossein worked on the membrane interface engineering. Yanyu Samuel and Mushumi work on the synthetic cell secretion. And the work I presented today had been a product with collaboration with a number of groups. And I want to thank our funding sources as well. And thank you so much for your attention, and I'd be happy to answer any questions.
Thank you. Thank you so much. That was awesome. There is a lot of questions in chat. Can you see chat?
Oh yeah, let me stop sharing so I can bring myself up to the okay.
If you can read a question out loud, because people watching the recording don't see the chat.
Yeah, absolutely, absolutely. Okay, so let me start. Let me start from very top. Questions says, could you please speculate how liposome compositions like membrane rigidity will affect cell skeleton? Yeah. Is there any design consideration for lipid composition we need to keep in mind? Oh, great question. So most of the study I've presented so far, we have not varied compositions very much. But certainly, I've done a bit of work myself during my PhD on membrane compositions. So by changing composition, you can alter rigidity. From a cell skeletal standpoint, it will change the deformability of the GVs. Now, bending rigidity with synthetic lipids, it's not you can tune it over a very large range. So what we typically work with is changing tension in the membrane by changing osmotic as a way to change the deformability of the membrane rather than using composition. Now, composition can play a role when it comes to like even membrane fusion. With the membrane fusion study I presented, we have to use a certain amount of PE, phosphoethanolamine lipids, and so that plays a role in those studies. So for the cell skeleton, we basically just have a very standard DOPC cholesterol membrane. So we haven't actually done so much with composition there. But depending on what you're trying to reconstitute, yes, lipid composition is a key parameter to play with, right? So hopefully that helps, but I can expand on any of these points later if there's a need. So the next question is, what is the most productive ratios of sender and receiver cell for highest yield in receivers? A great question. Um, Gosh, yeah, I think we just do an equal mixing of the two, and we rely on random chance of forming the interface. I don't think, Hossein has graduated from my lab. I think he would have been here for longer if we had to explore the racial part. Um, yeah, so I think this is just based on random formations of center receiver cells. If you imagine having more of the center cells and less of receiver cells, then it's, you may not have as many contact. And this is a sort of a fishing experiment that we did with mixing the two and hoping that they come in contact at a reasonable high concentration so we can find the interface. So I guess I don't have a clear answers on the productive ratio here. The one idea that I had before was there are some microfluidic system that allow you to form these cell pair, and maybe we could use it as a higher throughput method of creating, interface between sender and receiver cells, and that may give you a better chance of observing these, interfacial communication. All right, so I hope that helps. So there are a lot of questions. Maybe I'll go faster. The next one is, does calcium affect liposome stability? Is the working concentration of calcium low enough to not affect liposome stability, and are you using lipids that don't care about divalent ions? I'm not sure if about the stability part. So we use anywhere from 2 millimolar to 5 millimolar, and actually we do know that when you have very high calcium concentration, it's known that calcium can induce its own I guess fusion properties. So we try to work at, if possible, try to work at a low enough concentration that doesn't do that. I'm not sure about affecting liposome stability. At least in our case, in our hands, the stability is reasonable. We don't see ruptures of vesicles. And yes, the lipids do interact with divalent ions, so I guess including the P here is actually important for fusions. That's a common recipe for membrane fusion studies. So yeah, I don't know if I addressed that one well, but sorry, let me try to get some more. So next question is, how can we control when and where synthetic cells make contact to reduce randomness, better study the signaling behavior? I asked in terms to reduce heterogeneity in signaling outcome and to enable quantity analysis. Yeah, so this is a question that relates to the two questions earlier. Is right, at least in the study I presented, we don't really control the when. I guess the when is when we mix the two together. The where is definitely not a parameter that we play with. However, if you have this microfluidic trap system, then you may be able to position a population of center cells and the population of synthetic receiver cells in the same regions, and we can then build this signaling in a more controllable manner. Now, the other possibility, and this is just off the cuff, is maybe it's possible to create these, populations of center cells that are in a tissue. I know Higgins Lab is here. Think about these, synthetic cell tissues. You can maybe have a center cells that are in the core, and then maybe deposit the receiver cells on the periphery, and maybe that's a way to create these spatial signal that propagates outward. Maybe that's an idea that can be explored later on. Um, um, okay, so the next is an okay. It says naive questions. If you have a way to dissolve liposomes with light to release TEF, why not just trap copper? Yeah, exactly. Yep. So let me finish reading it. Yeah. If you have a way to dissolve liposome with light to release TEF, why not just trap the cargo inside the liposome and then degrade it with light to directly release the cargo instead of using the middleman of TEF? Absolutely. So this we have thought about this as an alternative approach. Is at least in the grand world that that this would be an easier way to achieve by just encapsulating the UV degradable lipid together with a cargo. So we could do that. I guess my other point is that we're trying to build a system that has a secretion that mimics the exocytosis process. So we kind of think it would be good to not break the plasma membrane, so to speak, of the synthetic cells, but have the a small vesicle be releasing something and then allowing the cargo to release. So that may be the only reason why we came up with this idea of secretion rather than just breaking the membrane to release. Um, the next question is, when you plan on life-cell interactions, are you worried about potential cytotoxicity of TXDL inside liposomes? Uh, yeah, absolutely. So I don't know. I don't have a good answer for that. I think this is something that reviewers from from grants would ask. And something I do want to I do think about is what are the safety concerns with using synthetic cells in a and together with a living system. I think we will have to study whether the TXTL content would have any adverse effect on the cells. But there are ways that we're working on to create this live cell synthetic cell interactions, and I think um, that is not toxic. And then maybe that ties back to the question from Hagen's lab is if we don't disrupt the synthetic cell membrane, maybe we can fully contain our TXDL system inside without being outside of without interacting with the living system. Um, oh yeah, thanks, Taishu, yeah, for the comment. And then let me move on to for your tox session two, as the protein are only binding at the interface of the SUV, how did you achieve the communication between different compartments? What information um, did they change? Okay, so um, let me see if I can process that. Hang on a second here. Yes, yeah, okay. Right. So maybe let me just, I think. Okay, so maybe it was just a clarity issue. So for the second part with interface engineering, all the proteins are on the outside. And I guess I skipped one piece of data that talks about the light it's generating when you add furimazine. The light is generated at the membrane-membrane interface in that very small gap of the two membranes. And that blue light can actually is strong enough that it crosses the lipid bilayer, the 5 nanometer lipid bilayer. And the eyelid is on the inner surface of the receiver cell. And the blue light is then able to activate the eyelid, allowing it to bind to SSPB. So um, that's the sort of the signaling mechanism we propose, right? Um, I'm, I hope that was the question. Yeah, I think that was the question. Okay, let's see here. What is your view on how external mechanical signal like shear flow or different matrix stiffness can be converted in quantity into the quantity that the mechanical channel sense, i.e. membrane? Great question. Thank you for that. So I actually did not mention the way that we envision membrane tension increases actually through shear flow. So a number of years ago, I worked with a theoretician to estimate whether shear flow from blood alone can activate MSCL versus staying bound. And I actually didn't even mention that. So, uh, the idea that I propose is that for synthetic cell to bind to it could be epithelial cells, or I say epithelial cell would be interstitial flow, but to say endothelial cells, there would be blood flow. If you just have the vesicle that flow within the shear flow, that alone is not sufficient to activate MSCL because the vesicle is not gonna just think about the blood cells or think about platelets that get activated by shear flow, or it doesn't get activated by shear flow, but it has to stay bound to a matrix. So the idea here is that the vesicle need to be bound to a substrate in order for shear flow to be able to act on the synthetic cell to deform the vesicle in order to increase membrane tension, right? So think about it as if you are flowing in a river. If I don't grab on anything, I can just flow in the lazy river without feeling any strain. But if you grab onto something and the current is still going, then you will actually still feel the force that's pulling you away. So the idea is that in order to increase membrane tension, you have to be bound to something, and then and the flow has to go over. So that was the idea. And then, oh, the last question. Okay, well, we're right on time. Does mTOR or other fluorescent protein break under force in the F-tractant system? Is that? Wait, I guess I'm not sure the last part of it.
Maybe that's actin misspelled.
Oh, okay. I
That's
see.
my guess.
But I didn't have the actin with M Cherry. Well, I guess M Cherry itself doesn't. Oh, maybe he's thinking about the case of reconstituting a split protein. I don't know. Well, I mean, the protein itself is not going to break under force. I guess that's maybe that, um, unless you completely unfold a fluorescent protein. FPS. I guess also don't know what FPS. Inactin cell skeleton. Oh, okay. I guess I'm still not fully for fluorescence protein. Um, hmm. Yeah, it's okay. I don't fully follow if I so fluorescent protein in the actin cell skeleton case. Um, the fluorescent protein itself wouldn't break under force if I were to say if you asking whether the actin system that generate forces on the membrane and its fluorescent attack are fused to a fluorescent protein that oh uh oh okay, and then figure two of the 2000. Oh, okay. You're talking about the last part of it. Okay, now I think I understand what you're saying. Yeah. Sorry, I was thinking the first part. So there's no force involved in the last study. Now, I don't see any reason why force would allow the fluorescent protein to break. The release mechanisms based on protease cleavage and yeah and yeah and I don't suspect the force would have impact there if that's what you're asking. Okay. Um, all right. I think I got
All
through it,
right.
right?
I think you got through all of it. Yes,
Okay, cool.
I really like the questions. I was really interested
Sounds
in
great.
myself, particularly in the divalent current stability question, because
Mm.
we had some of that problems like that with magnesium, not calcium.
Ah, I see. Okay, okay. Interesting. So do you see a weakening stability with magnesium? Is that
They get, yeah, especially for like POPC cholesterol vesicles, we see that
oh,
if we
which is why
have
it
a
moves a lot.
too much magnesium, and by too much I mean like tens of
Oh,
millimolars.
tens of millimolar. Okay, I see. I see. All
Because
right.
I wanted to do RNA ribozyme reactions.
Mhm.
Those guys are really magnesium hungry, and we had to balance really carefully. The liposomes get leaky.
Mhm.
They almost kind of like
Yeah,
the lipids almost like precipitate at high magnesium.
I see.
And so
Okay.
we had to
Right.
balance the
Yeah.
concentrations, but we were nowhere near the concentrations that you're using. I mean, you have
Yeah, like in the
safe,
low, yeah, in the low minimal, right, right. And then yeah, exactly. Yeah. I think the blood has a couple, low millimolar calcium, which is why we settle on that. And also I guess, as most
yeah.
of you know, you know, um, calcium is a very conserved secondary messenger, right? Uses very heavily in signaling. So kind of start with the simplest signaling mechanism by leveraging calcium signaling, right? Yeah. So there's a lot to
Mhm.
do with calcium, I think, if we're going to be creative about it. So yeah, great. It's
All
at one
right.
o'clock. I'm not sure what is the
All right.
protocol here for you
Mm.
to call it
I
or
think I'll just,
yeah.
yeah, I think I'll just call it unless anyone else has any other questions. Going once, going twice. And thank you so much, Alan. And
Well, thank
thanks,
you, Kate. Well,
everyone.
all right. Thanks,
And have a
everybody.
great rest
Bye
of
bye
your
again.
Monday.
All right. All right. It's okay. Good to see you all. All right.
Bye.
Yeah. Bye.